Here, the types of introns. As we said, the introns have many functions. Based on its types, its function varies. These introns can be anti-ladder or anti-trailer sequence, promoter sequence. This promoter sequence are very important during the transcription process. Enhancer is going to increase the transcription. Pseudogenes, these are false genes. Gene fragments or telomeres. Telomeres were very important for the stability of nucleic acid at the end of replication. And a non-coding functional RNA. It can be a non-coding functional RNA. It can be assistance regulatory elements, which is going to very important to control the gene expression. So we will see about each of these types of introns individually. First, the anti-leader anti-trailer sequence. As anti-leader sequence is going to be present at the 5' end. At the 5' end, before the gene which is going to start for coding, there is a certain sequence of tandem repeats. It is known as 5' end. This 5' end is known as anti-leader sequence, which is going to lead the upcoming genes. And 3' end, which is the end of the DNA. End of DNA, which is going to consist of repeats of units of introns. This repeat of introns at the end of the DNA is known as anti-trailer sequence, whereas the 5' end, which is going to consist of a repeat, which is a start of a DNA, is known as anti-leader sequence. Anti-trailer sequence is the end of the DNA repeat. These 5' end or 3' end are not going to be translated, but it is present in the DNA. This 5' end is going to be between the transcription start site and the initiation codon. A transcription start site is also an intron, which is not going to involve in coding the DNA, coding the RNA, where the initiation codon is a coding, first coding RNA. So, for the starting, before the starting region, there is an anti-leader sequence present. And 3' trailer sequence is at the termination codon. After the termination codon, there is a sequence of DNA nucleic acids which is going to be not coding for any of the proteins, but at the end. This is known as a repeat of units known as trailer sequence, anti-trailer sequence. Therefore, based on its location, if it is present at the 5' end, it is known as anti-leader, which is going to be a lead. Where it is present at the 3' end, an intro is going to be present at the 3' end is known as anti-trailer, because it is at the end, which is after the termination codon, where anti-leader is going to be before the starting site. Here in the image we can see that at the 5' end, before the transcription start site, the anti-leader sequence is present. That is at the starting of the region. Whereas at the 3' end, after the termination codon, the anti-trailer sequence is present. Based on its location, these types are known through his name. Next, a promoter sequence. A promoter sequence is very important during the translation process. For the initiation of the transcription, process, for the initiation of the transcription, these promoter sequences are very important. A promoter is a region of DNA that is involved in transcription at particular gene. Because during the transcription, at first the transcription elements or enzymes are going to first of all bind to the promoter sequence, because a promoter sequence are first found by these transcription elements of enzymes. So the transcription factors are first of all going to go. Here we can find that this is the RNA polymerase, which is the enzyme which is involved in transcription process. The RNA polymerase are going to convert the DNA into RNA. So, the RNA polymerase is going to produce the RNA from the DNA. For that, it first of all binds to the promoter sequence. here we can find this is a promoter region. Minus 35 and minus 10 region is a promoter region. First of all, the RNA polymerase is going to first of all bind to these promoter sequence. After that, only it will move on to transcription start site. After that, it will binds to the region of first coding region. A first amino acid to be coded can be synthesized after it reaches a promoter transcription site, and then only it moves on to the first coding region of gene. It will reach exons. So for the directing the RNA polymerase, these promoter sequence are very important. If the promoter sequence is not present, then the RNA polymerase cannot find the DNA which is going to be coded into RNA, which is going to be converted into RNA. Because therefore the promoter sequence is very important. They are located in the upstream of the gene, which means a gene before a gene which is going to be coded, before that there is a certain types of nucleic acid is known as upstream of a gene. So a Tautor box in general, a Tautor box in prokaryotes is known to be of 25 to 35 base long, is going to involve in acts as a promoter sequence. This Tautor box is going to direct the RNA polymerase binding, which is then directed into the transcription process. Therefore, promoter sequence is very important. This promoter sequence is also a repeated units of 25 to 35 bases long, is very important for the transcription process.